The overall goal of this procedure is to detect low-abundance growth factor receptors in zebrafish embryos by combining the selectivity and sensitivity of affinity labeling of the chosen receptor by its radioactive ligand.
This method can help answer questions in developmental biology fields, such as receptor expression levels and their biochemical properties.
This method, which we have nicknamed AFLIP, extends the powerful descriptive qualities of affinity labeling, a well-established cell biology technique, to zebrafish developmental studies.
To begin this procedure, collect 100 to 200 embryos for each condition and place them in petri dishes containing fish water. Then, manually decorionate the embryos using fine-tipped forceps and avoid using pronase or any other protease throughout the protocol, as it may digest the targeted receptors. Next, place the embryos in a 1.5 milliliter microfuge tube and wash them twice with 1x PBS. Then, add 500 microliters of de-yolking buffer. Release most of the yolk by gently pipetting the embryos in the solution up and down about 30 to 40 times. For 72 HPF and 48 HPF embryos, use blue and yellow pipette tips respectively. Yellow tips may need to be slightly cut to avoid destroying the embryos. A successful yolk release can be checked by observing the embryos under the microscope.
Removal of yolk must be as complete as possible, as yolk, lipids, and proteins may interfere with subsequent ligand or antibody